Hello, it's my pleasure to be here, and I'd like to thank the organizers for the opportunity to present our work today. My name is Julio Saez-Rodriguez. I'm from Heidelberg University. Before I start, I should disclose funding from GSK and Sanofi and fees from Traveler and Astex. With that out of the way, I'd like today to present some of our work where we try to combine biological prior knowledge with multi-omics data, in particular single-cell multi-omic data, to understand cellular regulation, in particular in the context of human disease. The question that many of you are also working on is how we can analyze large multi-omic data sets like proteomic, transcriptomic, and so forth. So this is data we collect for a large number of things across different samples. These samples can be coming from the laboratory, or in our case, where we're interested in using this omics data to better treat patients, they may come also from patients. And then also with development in the recent years, we can measure this not just in a bulk manner, so a particular gene in a particular sample, but with the resolution of each individual single cell in that sample, or even with information about the spatial location of the cell. So all these large data sets, that we can use them, as we said, to do different types of analysis. In our case, in particular, we try to understand human disease and better predict which therapies will work on which patient. The general question then is, can we use statistics and machine learning methods to find these large omics data sets, patterns that are perhaps associated, let's say in our case with the development of a disease, or can we predict, for example, if a drug is going to be efficacious for a given patient? So what I want to tell you today is our work on how we use biological knowledge to support this process. And the idea is that rather than bringing the data directly into your favorite machine learning algorithm, you would before extract signatures. The signatures, as I will show you, are, for example, activity of a transcription factor, are particular features that are informative of mechanisms, and they are stored from the omics data and biological knowledge. And the value is twofold. On one hand, we reduce the dimensionality of the data because the number of features is less than the number of genes, so maybe we have a few dozens of transcription factors instead of thousands of genes. So this increases the statistical power of our approaches. But also, and importantly, these signatures are rooted in biochemical processes, so they have a very direct mechanistic interpretation, and also they are very amenable for follow-up experiments in terms of validation. So all the tools and methods and resources I will show you today are free open source in our website. Visit here also our GitHub repository, and we're really happy if you use them and to welcome feedback and suggestions about them. The first thing that we need if we want to use biological knowledge is to have an easy way to access it in a cohesive manner. For this reason, we have developed over the years OmniPath, which is just a one-stop shop which gives you access to over 100 different knowledge resources. So this started in our case with work around biological networks and pathways, then with different types of information, such as the aspect of different enzymes, molecular complexes, annotation about localization and other things, as well as information about localization and communication between different cells. So this information is available for over 20,000 proteins, and for us it's quite important that we have also annotations about these proteins, because this is informative and something we can also look into more detail, know more of what's happening in a particular process. So all of this is freely available through R, Python, and a Cytoscape plugin. And really the basic idea of OmniPath is that in the same way that if you wear glasses, you would go to the ophthalmologist to see which is the best lens that you can see to see the best. With OmniPath, you can try different resources and find the best resource or maybe combination of resources to analyze your data with a particular. And because there are many other domains and many other databases out there that can be helpful, in the process now that we are going to refactor OmniPath as a graphical database, using Neo4j, we are trying to develop a unified language, a unified graphical database that should allow us to combine knowledge from different domains. For example, besides OmniPath on the molecular processes, targets, information about genetics and drugs, Intra, which is some text mining, or also Crossbar, which is more in the chemical side, and then this is work in the lab led by Sebastian and Dennis. So now I go back to our use of this knowledge for analysis. So as I said at the beginning, what we try to do is to extract signatures from omics data using biological methods. And what we have found, which is very informative, is to think of the omics data as the footprint of a process of interest. So oftentimes we want to know what's the activity of certain molecular players, such as transcription factors, but these are hard to measure at high throughput and with large coverage. But we do know that these processes affect some omics data, and then by measuring the changes in the omics data, we can estimate the activity of the process of interest. So for this, what we need to know these causal links, how the process of interest affects the omics data, and this is what we obtain from prior knowledge. Let me illustrate this in some cases, which I think will become more clear. So if you have transcriptomic data, you can use it to estimate the activity of the pathways by looking at the target. So these are the genes that change when a pathway is activated or not. So, and as depicted here, if you have here a particular pathway of interest going from the receptor to different proteins, all the way down to the transcription factor in the nucleus, you could on one hand say, okay, I will look at the expression of these components, and changes in their expression, I would assume, will tell me about changing the activity of the pathway. But we know that pathways are basically mainly constituted by proteins, and that the changes in the RNA level does not necessarily translate directly in changes in protein levels. And even more, that the activity of these proteins depends not only on their level, but also, for example, in particular modifications such as phosphorylation or the localization in the cell. For this reason, we and others have found that in this case, looking at the footprint of the pathway, the target is more informative of its activity. Along the same lines, we can look at the footprints of transcription factors to estimate their activity. So in this case, what we do is to look at regulars, so information about target genes of transcription factors. For this, there are great databases out there, such as Jasper and others. We have brought them together in one resource called Rothia in the lab. And then the idea is then again that we don't look at the changes in the expression of the transcription factor itself, but the changes in the expression of the target genes to estimate its activity. The same idea can be applied to other types of omics. If, for example, you have phosphoproteomic data, you can use it to estimate the activity of kinases, because the kinase drives the changes in the phosphorylation of proteins. Or if you have metabolomic data, you can use it to estimate the activity of metabolic enzymes, which are also controlling the changes. How you would do this estimation of the activity from the target is something you can do with many methods. And because we wanted to be able to try different ones and combine them, so we developed a small package called decoupleR, which is a collection of these enrichment methods that can be applied to any prior knowledge and any type of data. And then you can use methods from overrepresentation analysis to different variations of instant enrichment analysis and many others, as well as a consensus method. And this is an R package, also a very fast Python package, and also is available through a Shiny app. So until now, I told you how we extract features from omics data. We're also very interested in how we can integrate these features in a network context. And for this, we developed a methodology called Carnival, which tries to find causal paths in large networks that connect upstream drivers, such as changes in receptors, with downstream changes such as transcription factors. And for this, we use linear programming, which allow us to very efficiently find paths in very large networks. More recently, we have expanded this idea of causal paths to multi-omics. And this is a tool called Cosmos that allows a causal integration multi-omic data with prior knowledge. And the idea then is you will have kinases, transcription factor activities, metabolites, and trying to find links in large networks and combine them. Let me tell you a bit more about this, and I will use an example to illustrate this methodology behind Cosmos. So this is work on kidney cancer we did with Raphael Kramann's lab in Aachen, Christian Fritza in Cambridge at the time, now in Cologne, and Jesper Olsen in Copenhagen. And our experimental colleagues analyze tumor and matching healthy tissue of kidney cancer patients with mass spectrometry and RNA sequencing. So we obtain phosphoproteomic, metabolomic, and transcriptomic. Then first from these different omics, following these ideas of the footprint, I just illustrated, we estimated the activity of transcription factor, kinases, and metabolites. And then using Cosmos, we try to connect them in these large networks. So what does this look like? So here on the left, you can see the starting network is a very complex hairball with 117,000 interactions. This comes largely from OmniPath, our meta resource, doing many signaling related networks and pathways. We combine this with Recon3D, a large metabolic network, as well with Stitch, a databases of the binding of metabolites to different proteins. So when you map on this network, the kinase, transcription factor, and metabolites, and then you try to find with the algorithm the interactions among them, and then you obtain a network such as the one on the right. And as you see here, then the network is fairly complex, but certainly much less so than the one on the left. And then you can zoom in into specific parts of these networks and look at specific mechanisms that bridge from metabolism, signaling, and gene regulation, like here, for example, metabolic enzyme, and then the corresponding metabolite affects a prescription factor, and so on. But here we have noted with numbers specific mechanisms, some of which, when we look in the literature, they were supported, they were known to happen. Some others were not known. So these are new hypotheses that can be then further validated. And I want to emphasize that these methods, such as many others in our field, are really hypothesis generation tools. So we have data with limitations, biases, noise, and so forth. In our second input, the biological knowledge is also incomplete. And then we also have assumptions in our algorithms. So these methods may or may not give us the right answer, but at least they give us hypotheses to follow. Let me now summarize this first part of my talk. So I try to explain to you how we use biological knowledge to extract signatures from omics data, and how we focus on this idea of the footprint, that if instead of measuring something directly, you can estimate this activity by looking at the downstream effects, which are easier to measure. And then how we can use this together with large networks to identify causal paths that connect them and develop mechanistic hypotheses about processes of interest, and that you can do this using different omics, phosphoproteomic, transcriptomic, metabolomic, and this is something we are also extending to other types of data as well. Good, so far I've been talking really in the context of bulk data, but as I said at the beginning of the talk, there is also, of course, a lot of single cell data, and one question that we had is, do these methods that work well and are developed for bulk data work on single cell data? And to cut a long story short, and this is now work published a couple of years ago, we found that at least in the context of our benchmarks, that is the case. And why? The basic idea is simply that if you are looking at let's say, single cell RNA data that is great because you have resolution of individual cells, but it has limitations, for example, not complete coverage of all the transcripts as opposed to bulk. Still, you may get enough information to estimate the activity of the processes of interest because the footprint is composed by multiple genes. So you don't need to measure them all. If you measure a few, it can be enough to get the output or information about the process. So what this means then is that we can apply these methods to single cell data. This is just one example. Collaboration with Raphael Kramann in Aachen. They were looking at kidney fibrosis. So they were looking at using single cell RNA, and we use PROGENY, which is a method to estimate the activity of pathways, looking at the footprints. And they were in particular looking at different cell types, myofibroblasts, fibroblasts, and pericytes, because these are the major producers of intercellular matrix. And there was a particular subtype that had interest in increased extracellular matrix production, these NKTB plus cells. So using these pathway methods, we found that activity of TGF-beta is higher in NKT2 positive cells. And this made sense in the context of fibrosis, because this pathway is a major driver of fibrosis. And then this allowed to characterize the cells, and together led to the validation of this subtype by a knockout of this NKT2+, which led to the decrease of extracellular matrix and accordingly TGF-beta activity. So just a small illustration of how you can use these methods to look at a particular group of cells with single cell resolution. So what then this means is now, as illustrated in this example, we can look at particular cell types and estimate pathways, transcription factor, and so on from RNA. And for this, we can use the tools I saw you from our group, but through OmniPath, we also provide our comprehensive knowledge. We can use other tools, such as, for example, ISMB, which is broadly used in single cell. It's a very powerful method to combine upstream ligand receptor signaling with downstream changes in transcription. And one type of analysis that is very common in the context of single-cell RNA is to look at cell-cell interactions. And the idea is that if you have two groups of cells, let's say different cell types, cell type A and B, if there is a high expression level, cell type A of a particular ligand and cell type B of the corresponding receptor, this is information that you get from databases and is also available through OmniPath, then there is a higher chance that these two cells are communicating, because one has high ligand and one has a high corresponding receptor. But if you think about this from the biological point of view, there is actually a number of steps in between, a number of assumptions that we have to make to do this analysis. So if two cells truly communicate, what needs to happen is first, certainly, that the ligand is expressed in one cell, but then this RNA has to be translated into protein. And if it's, let's say, a cytokine, this has to be secreted outside of the cell, it has to diffuse, and then it has to reach the target cell, interact with this receptor, and then trigger a response. And only then really we can say that two cells are truly communicating. But all we can measure is really the expression of the ligand and the expression of the corresponding receptor. So because of these assumptions, and because there are many methods out there, we developed a resource called LIANA, which stands for a ligand receptor analysis framework. LIANA is just a place where we brought together databases of cell-cell interactions, 16 different ones through OmniPath, as well as 8 existing methods such as CellPhoneDB or CellChat that allow us to look at these cell-cell interactions. And we also provided the option to complete a consensus across the methods. So then if you have your single-cell RNA data clustered in different groups, then you can use it to estimate how cells interact with any of these methods. And because there are so many methods, we try to see how they perform. First, what we found in several cases that both the method and the underlying research we use can influence the results. And for this reason, we recommend to use consensus of different methods. And second, we try to really benchmark, and we struggle to find really definitive ground rules. So cases where we know that two cells are really communicating and you have single-cell RNA data for them. And because we couldn't find it, and this is something that if anyone has good ideas, we're happy to discuss, what we could do is indirect benchmarks. So for example, you can look at the signatures of the target cells, the cell that receives the ligand, do the downstream genes, downstream of the corresponding receptor change, or we also use site-seq data where you have protein level of the receptors, as well as spatial transcriptomics. And the case of using spatial transcriptomics is that we thought that if cells are communicating, it's more likely that they are next to each other, or the other way around, if they're next to each other, it's more likely that they are communicating. And of course, it will not always be the case, but there should be some correlation. And indeed, that's what we found for spatial transcriptomics, but also for the other benchmarks, that in general, these methods capture signal, And this is illustrated here, where we look at spatial transcriptomics on brain cortex and triple negative breast cancer. And here, this parameter, whenever it's above these dashed lines, means it got signal, it was better than random. And as you see, most of the methods in most of the cases. So, and the specific results change from case study to case study, but in general, these methods do give you positive signals, or give you hypotheses, but of course, there are many assumptions, so these hypotheses have to be further validated or studied with other strategies. So that's some vignettes on approaches to study signaling on single cells. But there is, of course, an important aspect that we cannot look at when we have single cell data, which is the actual spatial location. Because the single cell methods, what they do is to dissociate the cells and then analyze the sequence. But if these cells are coming from a tissue, it does matter a lot where was the cell in the tissue, who were the neighbors, because neighbor cells are presumably coordinated in a particular. And as you probably know, over the last years, there have been amazing developments, and as we speak, there are still many technologies being developed and coming out to be used that allow us to measure RNA as well as proteins with spatial resolution. So we were quite interested in how we can use this type of data, and in particular, we work in the context of studying the human myocardial infarction to use spatial transcriptomics combined with other technologies. So this is work led by Christoph Cooper, Ricardo Ramirez in our lab, and me. So Christoph is from the Kramann lab in Aachen, and Cindy from Costa's lab in Aachen, and we also collaborated with Milton. So what our collaborators have access to is to samples heart failure patients, meaning patients that suffer an infarction, and for them we had samples in the ischemic zones where the infarction happened, as well as distant zones and somewhere in between the superborder zone. We had healthy controls, and as well as chronic patients. And in all of these samples, we had done perform spatial transcriptomics using the vision technology. So this is a really powerful technology, which very good in coverage, but it doesn't truly have single cell resolution. So each of the spots that you measure in the tissue, so you know where they are, but they are of a size that you would typically have 5 to 10 cells in each of them. For the reason what we've done, and what also other groups do, is to at the same time from a sample next to the one where you do the spatial transcriptomic, you do this through single cell sequencing, in our case nuclear RNA-seq, with the idea that then you can combine them both, you can improve the resolution of the spatial result data. And in addition, we had attack 6 information about chromatin accessibility of the sample. So then the idea is how we can use computational methods to integrate the spatial data, the single cell data, and the attack data to understand the regulation in the human heart in the context of myocardial infarction, both intra and intercellular. So for this, there are different analyses we perform. One thing that you can do is, as I said before, because the spatial transcriptomic does not have single cell resolution, there are methods to use this accompanying single nuke RNA-seq data to estimate in each spot the amount of cells. So we use in our case a method called cell to location that allows you then to estimate, and then you can see if you have particular cell types which are relevant for the heart. But also in each of these spots, we can leverage these tools I presented before to extract footprints, to extract signatures, which again would allow us to have a higher statistical power and a higher interpretation. So this is one example of that. This is one sample of a patient suffering chronic myocardial infarction. And with these methods to estimate the cell types, we kind of knew that there is a large number of fibroblasts of a particular type, fibroblast 1 in this central area, and in the border, fibroblast 2. If now in each of these spots you compute these footprint signatures, we saw that the activity of TGF-beta is high where these fibroblasts are present, something that makes sense because fibroblasts drive fibrosis, and TGF-beta is critical pathway in this process. We could also look at downstream transcription factors such as ETF4, again, is active in similar locations and the corresponding downstream regulated genes. And here is where we use the attack data to refine our information about the transcription factor targets, about the gene regulatory networks. And the reason being that if you use the resources, like I mentioned before, they are very helpful, but they are largely not cell type specific. But using the attack information, you can get cell type specific knowledge about how the chromatin is open, and therefore you can use this to better refine your gene regulatory network. And this was worked on with the Costa lab. And because this is a very interesting development in the field, we were considering which methodologies we could use. And what we realized is that there is a large time of methods out there already to infer gene regulatory networks from RNA and attack. And here are listed some of them, but there are really many more. And if you look at them, all of them are somehow different, they have different assumptions, probably different aims, and therefore we were wondering again which method to use. And so Pau Badia, a student in the lab with Lorna and Remy, and in a project which is also in collaboration with other labs, in particular Judith Sauk, Karl Hermann, Steinertz, and even Saez, is developing a framework called GRETA, which stands for Gene Regulation Network Analysis, which is a framework where we can analyze different methods to estimate gene regulatory networks from RNA and attack, evaluate according to a number of metrics, and also perform different type of analysis to see what these methods are telling us. And this is work in progress, and we really welcome anyone who wants to join us in this effort, because we think it's something that would be good to develop as a community. So until now, we have, coming back to the case, the project in the heart, we have a look at individual spots and analyze them, but really not use the fact that we know the different location of different spots. This is can be very informative, because we can find not only spatial patterns, but also use this localization information to estimate if particular cells, if particular positions in our tissue are interacting. And to help us analyze this type of data, we developed a method called MISTIC. So MISTIC is a multi-view machine learning framework where you can precisely try to do that, try to see how much of the changes that you find in one place in your sample. This can be a single cell if it's single cell resolution, or can be a spot with multiple cells if it's like in the case of the Visium. How much of the changes in that spot depends on the spot itself, how much depends on the closest neighbors, the local view, more distant neighbors, perhaps mediated by diffusion of different molecules, like what we call the tissue view, or really any other view that you want to apply, because the framework is flexible to include bespoke views. And we have applied this in different cases. In this publication, for example, we saw how this can be used to multiplex transcriptomic as well as proteomic data. So with that, I'd like to finish thanking first the people in the lab. I mentioned them along the way, but the key people of what I showed you today is Denise behind Omnipath, Aurélien, the multi-omics integration, Pau, the gene regulatory network analysis, Ricardo, the analysis in the heart, and Giovanni's MISTIC, spatial analysis. I mentioned our collaborators along the way. If you like what we do, we are always looking for motivated and friendly students and postdocs. And I leave you with take-home messages that I try to convey today. So I try to make this point that biological knowledge can be really helpful to support machine learning and statistics, analyze multi-omic single-cell data, improving the performance and interpretability. Omnipath, our one-stop shop for biological knowledge. How this idea of the footprint of the target genes it can be quite helpful to extract features from omics data. We really think benchmarking is really important. I showed you a couple of examples, but something I think important to the community also to do so together as part of crowdsourcing efforts is a very valuable strategy, and we're part of the Dream Challenges that follow this strategy. And finally, at the end, I showed you how spatial omics is really interesting, but there are also very cool challenges for computational people like us. With that, thank you for the attention. I'm happy to take any questions. All our resources are free, and we hope you find them useful. Thank you.